So we continue with lecture 6, where we cover the nucleus and the rough endoplasmic reticulum. Let's start with the nucleus. The nucleus is the Latin word for nut, and as I said, it is a very prominent structure within the cell that contains the genetic material in form of DNA or RNA. And I've given you an example here where you can see mechanosensory neurons, and these are the somata, the cell bodies of those cells. And here you can see that each one of those cells contains one very big prominent nucleus. And in general, this is the rule that each cell contains one nucleus. However, there are some cells, some exceptions. For example, the red blood cells don't contain nuclei, and platelets don't contain nuclei. And the likely reason for this is that they have to flow through this very fine capillary system within the extremities of organisms, and they would likely not fit through those very fine capillaries if they did have nuclei. The drawback for this is that they have a very limited time span for that reason. So for that reason, red blood cells and platelets need to be produced quite frequently, millions every day by the blood system in order to replenish the ones that die. There's another exception in the other extreme, and these are muscle cells. And muscle cells are multinucleated cells. And I have an example for you here. So this is a muscle cell, and you can see that there are multiple nuclei within this muscle cell. There are other examples for multinucleated compartments throughout the body, but these are the main cells. Not all nuclei look the same, and it depends on what kind of cell they are located within, and I'll give you a few examples. However, if you think about the number of different cells that we have in our bodies, we have neurons, we have bone, we have kidney cells, we have muscle cells, we have intestinal cells, we have all kinds of different cells, receptor cells, that all have different tasks, however, and express different proteins. However, they all have the same genome, they all have the same genetic material within them, and this is known as genomic equivalence. So theoretically, a neuron can express all of the proteins that a muscle cell can express, and vice versa. However, it would probably not be the best thing if your motor neurons themselves were contractile. For that reason, the motor neurons do not express proteins that muscle cells express, and vice versa. And the reason why they don't express these other proteins that are expressed in those particular cells is that they are down-regulated to certain mechanisms and mainly proteins that bind to those genes that should not be expressed in cells and silence them and downregulate them. But in principle, the genome in a neuron compared to a muscle cell is the same. So in eukaryotic organisms, the DNA within the nucleus is in form of linear chromosomes, and the number of chromosomes within the nucleus varies between different species. In the case of humans, we have 46 chromosomes. We have 23 pairs, makes 46 chromosomes. And this is true for all of the somatic cells, with the exception of the gametes, the sperm, and the egg cell. They undergo meiotic division, a reduction division, and for that reason they only have one chromosome set. So they only have 23 chromosomes within them. All the rest has a set of 2 chromosomes. All the somatic cells that make up all the rest of the body have a set of 2 chromosomes within them. So here's an example for how different nuclei can look in different cells. And again, I come back to my neurons, and this is a beautiful example here. If I have my mechanosensory neurons, you can see that the cytoplasm of these mechanosensory neurons is very big, and for that reason the nuclei are not compressed. So there are other cells like these glial cells, and glial cells are insulating cells that wrap themselves around neurons, pretty much like the insulation around a copper wire and an electric wire. So the insulation would be those glial cells that wrap around these neurons. That would be your copper wire in your example. And as you can see, the cytoplasm within those glial cells is very compressed, very tight. And for that reason, the nuclei within them are also very compressed. And you can see this here. So these are glial cells. I've stained them with a fluorescent dye, and it's called Hurst blue. And these nuclei are very compressed within these sheets of the glial cells. And for that reason, they look very bright because the genetic material is so compacted. In those larger neurons that look very similar to those ones here, the genetic material that is the same than in these nuclei here is spread over a much wider area, and for that reason it's much dimmer here because we have a bigger volume where the same genetic material is contained. And you can see here are slightly smaller neurons, and here the nucleus is somewhat in between in brightness between those little compacted nuclei in the glial cells, the nuclei and the intermediate cells, and the nuclei in the large cells. So depending on how much space this nucleus has, that's how compacted this nucleus is. And this is particularly fascinating and interesting in sperm cells. If you think about sperm cells and what their main function is they need to be very compacted, streamline shaped, almost torpedo shaped, and they need to carry this genetic material through in mammals, through the fallopian tube, to the infundibulum where the fertilization takes place, to the very tip of the fallopian tube. So those sperm need to be very efficient swimmers. And a big nucleus and not streamline shaped would be a disadvantage for those for the sperm. And if you look here at the seminiferous tubule where the sperm is synthesized in the testes of males, you can see these are the cells from which the sperm develops or differentiates. These are the stem cells here, and how big their nuclei are. And then they undergo meiotic division, so their genetic material is divided in half, basically. So we only have one chromosome of each chromosome, half the haploid set. And you can see how compacted the nucleus in those sperm cells that are in the lumen of the seminiferous tubule are here, is compared to the precursor cells from which those develop. And this is because the genetic material here is now wrapped differently around proteins within the nucleus. Initially in those cells, the DNA is wrapped around histones. These are specific proteins around which the DNA is wrapped, and we'll learn about histones more later. In sperm cells, the genetic material is no longer wrapped around histones, but here it's wrapped around proteins that are called protamines. And those protamines allow this genetic material to be wrapped much tighter, and for that reason be that streamlined shaped torpedo shape that enables the sperm to wiggle so efficiently. However, the drawback of this is that there's little gene expression that can take place when the chromosomes are so compacted. And for that reason, the main purpose of those sperm cells is to swim, find the egg cell, penetrate the egg cell, fertilize the egg cell, and form the zygote. And this is very interesting how nuclei are different in different cells. So the genetic material is surrounded by this nuclear envelope, which is a double membrane, and you can see this very nicely here. And this double membrane has these pores, and I'll show you an electron micrograph here. So if you have the double membrane here and you have those pores here, that's equivalent to this, you can see that those pores are very limited in size. So there are only small molecules that can be transported in and transported out of this nucleus. And these are small proteins, and this is messenger RNA that is transported, or mainly messenger RNA that is transported out of the nucleus for protein synthesis. The nucleus has also this nucleolus that I've talked about, and the nucleolus is the site where biogenesis of ribosomes takes place. So ribosomes, as we said earlier, are important for protein synthesis, and they are synthesized within this little electron-dense part here in the center of the nucleus. It is not surrounded by a membrane, but it contains high amounts of ribosomal proteins and ribosomal RNA. So those ribosomes play an important role in protein synthesis, and they consist out of two subunits, a large subunit and a small subunit. And these two subunits only come together when the protein is actually translated at the rough endoplasmic reticulum. So the messenger RNA is transported out of the nucleus and is then, depending on what kind of protein it encodes for, either guided to the rough endoplasmic reticulum or it remains in the cytoplasm. So if you look at this picture here again, this is our fluid mosaic model of the cell membrane, and we said that there are some proteins that are inserted into the membrane. And then there are some proteins that are surface proteins, or there are some proteins that are not at all attached to the membrane. So depending on what kind of protein is synthesized from the messenger RNA, those ribosomes now either translate the messenger RNA in the cytoplasm and form proteins that are not incorporated into membranes but are free-floating in the cytoplasm, or they are guided through signal peptides to the rough endoplasmic reticulum, where the ribosomes now translate this messenger RNA and now incorporate the protein that is produced into the membrane and basically form those integral transmembrane proteins that need to be incorporated into the membrane in order to allow controlled signaling from the inside to the outside, or from the outside to the inside of the cell. So two types of synthesis. One synthesis is free-floating in the cytoplasm, and the other one is at the membrane of the rough endoplasmic reticulum, where the protein that is now translated is directly inserted into the membrane of the endoplasmic reticulum. Structurally, those ribosomes are the same, and here you can see an electron micrograph, so you can see both of those ribosome types. So here you can see those membrane stacks. Here are the internal, the cisternae, the internal parts of this rough endoplasmic reticulum. And here you can see the ribosomes, these electron-dense dots that are on this rough endoplasmic reticulum. And they synthesize the proteins that are incorporated into the membrane. And here you can see free-floating ribosomes, and these ones are the ones that usually translate proteins that are cytoplasmic proteins and are not incorporated into the membrane. And this concludes our lecture on the nucleus and the rough endoplasmic reticulum.